Hello, 3 p.m. section. Hey, last couple of minutes that I ran out of time, just the last couple of points here. So, as I mentioned before I ran out of time, the metabolic diversity of prokaryotes greatly exceeds what one sees in eukaryotes. It's really quite extraordinary. And again, don't get lost in the details, but try to wrap your mind around the big concept. So, for example, your book does a terrific job of identifying two major things to focus on in considering metabolic diversity. One is, what is the source of energy for the production of ATP? And in bacteria and archaeans, we have three different ways. Light energy can be used. The energy locked up in the bonds of organic molecules can be used, or inorganic molecules. This last category may be less familiar to you. You probably recognize the light as a source of energy because this is, of course, is what plants do. And we animals oxidize organic molecules for producing ATP. So this is also familiar to you. These terms may not be. They're pretty self-explanatory. Troph means having to do with eating. And so light is a source of energy. Chemoorganic trophs are those organisms that are like animals that oxidize organic molecules, and chemolithotrophs, litho means having to do with rocks, are able to oxidize inorganic molecules. They don't have to be from rocks per se, but inorganic molecules as opposed to organic ones. The other way in which metabolic diversity is categorized is, where is the carbon coming from? What's the source of carbon for the production of organic molecules? Because after all, organic molecules have carbon in them. And autotrophs use carbon dioxide as a source of carbon. So a familiar analog, it would be plants. And heterotrophs are those organisms that can't produce all their own organic molecules from inorganic molecules such as CO2, but need a supply of at least one organic nutrient. So this is comparable then to what one sees in animals. Now, the sources of carbon and the sources of energy are completely different characteristics, so one can think about putting these as a 2x3 table, which was done for your convenience in the textbook. This is summary table 29.3, which is in front of you right now. So the columns here are the sources of carbon, and the rows are the sources of energy. We're more used to thinking of plants and animals, so I'd like to point out that plants would be in this particular combination here, where the source of carbon is the organic molecules are synthesized by the organism itself, so autotrophs, and but the energy comes from sunlight. So plants, for example, are photoautotrophs. Animals are chemoorganoheterotrophs. They're heterotrophs because they're using organic molecules as a source of CC bonds, and the source of energy is also from organic molecules. But in addition to these two major different metabolic lifestyles, which are characterized in the plants and animals, microbes have all 6 different combinations of these different combinations. So much more metabolically diverse, and they're able to carry out chemical reactions that humans and other animals are basically incapable of, to large part. I would encourage you to look at this information without worrying about memorizing it. This should amaze you by the virtuosity and chemical virtuosity of these organisms in terms of what kinds of molecules they're capable of using as electron donors and the kinds of byproducts that they can produce. So hydrogen and methane, for example, are not chemical compounds that eukaryotes are capable of using metabolically. I want to end by highlighting just one bacterial group for particular mention, and that is the group called the cyanobacteria, where there are pictures right here. Cyanobacteria are a rather extraordinary and important group. They are characterized by all performing oxygenic photosynthesis, oxygenic meaning producing oxygen. So oxygen is produced as basically a waste product. So down here in this picture, you can see little bubbles of oxygen being released by these mats of cyanobacteria. And so this was very important in the evolution of life on the planet because these were the very first producers of oxygen. And you may or may not be aware that the oxygen in our atmosphere is all of biological origin. So it was made by some photosynthesizer, and the cyanobacteria were the very, very first oxygenic photosynthesizers, and so they were the organisms responsible for the initial appearance of oxygen in the early atmosphere. Cyanobacteria can be solitary, as shown up here, meaning each cell is off on its lonesome, or they can be colonial, meaning that after a cell divides, they remain attached. This is different from being multicellular because each individual cell is completely autonomous and there's no differentiation in here. If we go back to the timeline, then the first evidence of oxygenic photosynthesis is right around the time of the first cyanobacteria. These should actually be coincident. I'm not quite sure why your textbook illustrator showed them as distinct because after all, it was a cyanobacteria that first started releasing oxygen into the atmosphere. And as it accumulated in the atmosphere, after long periods of time, hundreds of millions of years, then there starts to be a sufficient accumulation that geologists can see it showing up in rocks and so on.